Great. Well, hello to all the listeners out there. Welcome to our September Hordes Dairyman monthly webinar. My name is Kylene Anderson. I'm an editor for Hordes Dairyman. I will serve as your host for today's webinar, which will focus on what we've learned in the last 18 months of highly pathogenic avian influenza, or H5N1. If you're listening to the presentation live, you'll have access to a PDF of the slides that can be printed. The handout can be found in the handout section of the GoToWebinar control panel. In addition, if you have questions for our speaker, you can type them into the question section of the control panel, and we will address them following the presentation. Today, we welcome Dr. Jason Lombard to the Hordes webinar series. Jason earned his bachelor's, DVM, and master's in epidemiology at Colorado State University. He was a veterinary medical officer for USDA for 2 decades before becoming a veterinary epidemiologist for Colorado State, where he is today. Jason, welcome to the webinar, and I look forward to hearing your presentation titled H5N1 in Dairy Cattle, What We Have Learned in the Last 18 Months.
Thank you, Kylene. Thanks for everybody for joining. I'm going to go ahead and turn my camera off, and we'll get started. All right, so just to start off with a disclaimer, as Kylene said, I was with USDA for over 20 years, and the findings and conclusions in this presentation are my own and not construed to represent any official USDA or government policy. But some of the data that I will present today was made in part possible because of an agreement with USDA. Essentially, they funded a lot of our research. All right, so my aim today really is to describe the past and current situation of H5N1 in dairy cows, with an emphasis on exploring within herd and between herd transmission routes, including shedding patterns and duration in individual cows. So just a little background on influenza viruses. There's 4 types. Influenza A has been responsible for seasonal and global epidemics, and influenza B has been associated with seasonal epidemics. A and B are the influenza viruses that we as humans generally would get vaccinated for. Influenza C usually causes mild disease, no epidemics related to influenza C, and then influenza D is primarily found in cattle. And I wish we were talking about that today, but we're talking about influenza A, and it's divided into subtypes based on 2 surface proteins, the hemagglutinin, or the H, and the neuraminidase, or the N. So when we talk about H5N1, that's just referring to those 2 types of proteins. So the current outbreak in dairy cattle is really now gone from the H5N1 clade 2.3.4.4b genotype B3.313, and now has also included the genotype D1.1. So 95% of all farms that were infected over the last 18 months were with the B313, but then in Nevada and Arizona, we had D1.1. And the initial, well, I guess we'll talk a little bit about that in terms of transmission events. So just running through some quick historical events of H5N1. The first detection of this current outbreak was in February of 2022 in a commercial turkey flock in Indiana. But then early in 2020, and there were multiple backyard and commercial poultry flocks that were infected from 2022 through even the present. But in early 2024, we had this uncharacterized cattle morbidity event occurring in the Southwest US with reports of large bird die-offs and farm cats dying with neurologic signs. On March 20th, USDA confirmed H5N1 in goats on a farm with infected poultry in Minnesota, assuming that the goats got exposed to the infected poultry. That was the genotype B3.6, so not the B313 or the D1.1. A few days later, on March 25th, USDA confirms H5N1 in dairy cattle from Texas and Kansas, and that was the B313. That was thought to be a single-time spillover from wild migratory birds into dairy cattle. April 1st was the first human case reported in Texas associated with a dairy worker. Many of you probably saw the pictures of the conjunctivitis, the red eyes that dairy worker had. About a month later, USDA enforces the federal order pertaining to H5N1 in cattle. This really did 2 things. It required testing of lactating cows for interstate movement, and it required laboratories to report USDA either antigen or antibody positive results to state and federal authorities so that they could get a better handle on how far and wide H5N1 had spread. In USDA confirms H5N1 in alpacas on a farm with infected poultry in Idaho. This was the B313 strain, the same one as in dairy cattle. May 2024, again, FDA confirms H5N1 in dairy products is inactivated by current pasteurization procedures. I think that was really an important step to take to show that the milk supply is safe even with the H5N1. That same month, USDA confirms that the meat H5N1 virus in meat is inactivated by cooking to 63 degrees C. October 30th, USDA confirms H5N1 on a pig farm with infected poultry in Oregon. This was D1.2. Let's see, come on. December 6th, USDA begins the National Milk Testing Strategy Surveillance. We'll talk just a second about that. And then on February 2nd, 2025, genotype D1.1 was confirmed in dairy cattle. So really, I give you this as really there's a couple main points to take away. This outbreak has been in the U.S. for about 3.5 years. Multiple genotypes of influenza have been circulating in our livestock and poultry sectors, and most of the reservoir for this is wild migratory birds. And 2 genotypes have now been identified in dairy cattle. This is a map showing essentially the flocks that have been affected, both commercial and backyard flocks, since the February 22 detection. And you can see that it's been in all 50 states. About 1,700 flocks affected, 788 commercial, 921 backyard, 174.8 million birds affected. That really means most of those birds were depopulated due to the disease. I tried to upload this is from July, I tried to upload a new map yesterday to see what this had jumped up to, but the USDA website was running really slow and I wasn't able to get it. This map shows the detections of high pathogenic in wild and captive wild mammals from May of 2022 to present. And I think one of the things, when you look at that list of different species, bears, wildcats, domestic cats, marine mammals, raccoons, rodents, skunks, wild canine. There's a wide variety of mammals that this virus has infected. But I think the one that really stands out, I think, relative to the dairy situation is domestic cat, because that's one of the things that we heard early on was that the cats were dying even before a lot of farms saw any clinical signs in their cows. And now we've got some issues with some of the cat foods. You might have heard recently about a case in a cat that was infected with the virus, and these cats are really susceptible and usually die from the virus that came through cat food. All right, moving on to dairy. So this was a couple weeks ago. Again, 1,077, I think it's 1,078 confirmed cases now in 17 states. And with California and Idaho being the hardest hit and the most recent hit. The good news is that when we look at the last 30 days, there have been no new herds in any states that have been affected. So that's a really good sign that we've gone 30 days without a new detection. This is essentially a phylogenetic tree showing where the virus has kind of moved from state to state. And I think it's really interesting to look at all the places that it's gone, but then how those are kind of related in terms of New Mexico, Oklahoma, Minnesota, Colorado, all coming off of this branch up at the top. When we get down to this larger branch, we've got Colorado, Idaho, California, and Utah. So with some work looking at some of these sequences, we can get a better idea of how the virus is moving through our populations. But it's hit multiple states, multiple genotypes. I believe that, or not multiple genotypes, multiple strains of primarily the B313. And when we look at Colorado, I think when I talked to the folks that helped us with this, or put this together for us, there were 14 different strains in Colorado that arrived one way or another, but all through really, we think either through the movement of cattle or people not associated with an additional spillover from wild migratory birds. As far as the experts that look at this, they say that all the once that initial spillover event occurred in the southwest part of the U.S., now we can track all that through dairy cattle, sometimes dairy cattle to poultry, and sometimes potentially poultry back to dairy cattle. So the wild migratory birds really haven't been involved in this at a consistent level other than that initial introduction until we had Nevada and Arizona. Those were additional wild migratory bird spillovers into cattle. This is another graph that kind of shows, or figure kind of shows where it spread out of Texas, where it went. We know it went to North Carolina, went to Iowa, Michigan, primarily through the movement of lactating cows. And so the one of the things that we found out pretty quickly was that milk tends to have the highest viral load in it. And so the federal order coming out requiring the testing of lactating cows was really meant to stop the spread of disease from the states where it had already been impacted. And I think that strategy really worked well. We know we had an illegal movement from Idaho to California that really lit California up. But other than that, the federal order seemed to really shut down any new states that became infected. So the national milk testing strategy is essentially either silo or bulk tank testing in the states to try to make sure that we don't have more H5N1 that's circulating in herds. So 45 states are enrolled. This map shows those 45 states. It looks like North Dakota, Massachusetts, Florida, Alaska, and Hawaii are not currently enrolled. And then this shows the state status. So if we look at this, there's 24 states that are unaffected that are in the gold, and then the 18 states provisionally unaffected in the white, and then the blue states are still affected states at this point. All right, so what are the clinical signs in cattle? And some of you in these states will have a really good appreciation for all these, but the cattle that had some kind of either biometric sensor where they had rumen temperatures, we did see fever in almost all the cows that had clinical signs, and that was 4 to 5 degrees Fahrenheit above normal temperature for a couple days. And then what we saw after that was a severe decrease in rumination, in water intake, in feed intake, and activity. These cows had a depressed attitude. Initially, there was a lot of talk about the yellow, thick, colostrum-like milk and mastitis. That seemed to change a little bit over the course of the outbreak. And then what we saw primarily was more nasal discharge. And we were on farms usually a couple weeks after the onset of clinical signs. So this kind of shows what we saw when we were out there. This is some cows in the parlor. And you can see these, let me turn my pointer on here. You can see these, what looks like saliva hanging down. This is really all just mucus discharge, serous discharge from the nostrils, and almost all the cows in this line in the parlor had that. That usually progressed to kind of a mucopurulent nasal discharge, like these two cows. This cow had a really, she's a cow that was just detected as having clinical signs that morning we took the photo. This farm did use biometric sensors, so I think she was caught pretty early. But you can see her; she's got her head down, her ears are down. She's just like kind of looks depressed. She did have some nasal discharge as well. Some of the mastitis samples that we collected during our testing, not the thick colostrum milk at this point, but more chunky, watery milk with some blood in it as well. And then some herds we did see a lot of bloody nasal discharge. This cow was pretty sick. This cow had some bloody nasal discharge, but appeared otherwise healthy at this point. So one of the things that I think that this disease really was confusing to me, at least, is the variation in what it looked like from farm to farm. You'd go to one farm and they'd say, you know, we had it really mild. We didn't have any cows that died from it. And then you'd go to the next farm and they'd say, it hit us really hard. We had more than 20% of our cows infected, and you know, up to 15 to 20% of those died or were euthanized. So there was a lot of variety in the severity of clinical signs and the types of clinical signs that were seen in some of these herds. So one of the things that we can look at to measure how it's different in herds is the incidence rate. And so this is looking at 4 different herds in the green. Farm A and Farm BD both used biometric data. Farm A was the first farm that we worked with, and it looked like they had a nice, what we would consider kind of a nice epidemic curve. It looked like it was a single point introduction, which we do know that they brought animals in from Texas that was responsible for their herd becoming infected. And then it went up, it peaked, and this 4.16 essentially for an incidence rate means on that day 4% of their cows were detected with clinical signs. Compared to Farm BD, 6.5% of their cows on that day were detected with clinical signs. So we can see that it affected farms differently, from 3.4% of the cows at a high point up to 6%. And I think that some of the farms that use biometric data probably were catching cows that farms that didn't have access to that technology wouldn't have necessarily have caught. So at the peak of the outbreak, farms saw, like I say, 3.5 to 6.4% of lactating cows with clinical disease on a single day, which on some of these dairies is an amazing number of cattle to have to be attending to. And in most herds stopped seeing clinical signs between 25 and 35 days into the outbreak. But we did have some farms, Farm D here, for instance, they were still kind of seeing clinical signs out past 2 months. So this is an individual cow graph, and what we tended to see with most of the clinical cows that we looked at. This cow with the orange, that's her daily rumination. She went from 500 minutes a day down to about 50 minutes per day during her clinical disease event. And then milk production went from 100 pounds down to less than 20, and then she recovered about to 70 pounds. And so cows with clinical disease, it seemed like we had 3 different outcomes. One was a cow like this, where she did recover back to about 70 to 80% of her normal milk production. We had a percentage of cows that just completely dried off. Some of that may have been management based on their pregnancy status. And then some cows were euthanized or died, and that seemed to be kind of what we saw at clinical cows. We didn't see clinical cows necessarily that went back to their normal milk production in general. So this is Farm A again, about 3,600 cows, 22% of those had clinical signs. This is actually written up by Dr. Peña-Mosca at Cornell. We went back 7 months later. They had about 800 cows with clinical signs. Only 50% of those cows remained in the herd. A few of those were designated as do not breed cows. 8.5% of those had died or were euthanized, and then almost 40% were sold. So in some herds, this does have a big impact on both deaths and culling. So one of the things that we wanted to look at was what impact does this have on reproduction? And so in this particular herd, we looked at changes in the 21-day pregnancy rate risk. So we looked at it comparing 2023 to 2024. We know when the infected cattle were introduced. We know when the herd started seeing clinical signs. And so these asterisks essentially show when we have a significant decrease in our 21-day pregnancy risk during 2024. So for a couple 21-day periods, it dropped off, and then it kind of went back to where it was in 2023, and then it kind of dropped back down again, and we saw an increase in 2023's pregnancy risk. So there was definitely a significant impact to pregnancy in this herd. The other thing that we heard about was abortions. Some herds seemed to see a lot of abortions, and some herds reported no abortions. Again, looking at 2023 % abortions in the blue and % abortions for 2024 in the orange, we did see an increase. So this herd was infected in March and April, and we did see an increase in abortions in May. That's not typical for what we saw. Typically, we saw abortions right around the time of clinical signs in the cattle. But obviously, there's a lot of reasons for abortions. And we've had herds that had higher abortions in 2023 than they did in 2024 during their high path event. And so it's not a clear cause and effect for sure. So one of the things that through the experimental studies, they were able to show that the experimental injection of virus up into lactating cows could mimic the disease in experimentally infected cows. And when they euthanized those cows and looked at the mammary gland, they did see quite a bit of scar tissue. And so one of our concerns is, okay, well, these animals that recover from the disease, what's the impact on future milk production going to be? So this is Farm A again. So when we looked at next lactation, remember they had 800 sick cows, and 400 of them either were euthanized, died, or removed from the herd. We went back and looked, and so we used different days in milk cutoff because we know the further out we get in lactation, the better estimate we're going to have. But and this was the number of cows that we looked at in this analysis back in January. The good news is we saw in the 3 parameters we looked at, we saw about a 7 to 7.5% increase in peak milk in those cattle, a 16 to 17% increase in projected 305, and about an 8% increase in mature equivalent 305. So at least in this particular herd, and this is only one herd, we didn't see a dramatic impact on milk production in the subsequent lactation, but we've got multiple more herds to look at to see if this holds true. All right, so another thing we wanted to look at is what demographic of cows is this impacting in the herd? And again, early on we heard that this is affecting older cows that are later in lactation. So here, looking at it by lactation number, we can see that 16% of the cases were in first lactation, but 37% of the herd was in first lactation. So first lactation animals seem to be somewhat protected from clinical disease, but once we get out into second lactation and higher, we see a higher or very similar number of cases as the percent of cows in the herd. So H5N1 does affect cows in later lactation or older cows. When we looked at that by days in milk or days since calving, again, early on when we think cows would be most susceptible because of their immune status right after calving, they were actually at lower risk of having clinical signs. And it wasn't until we got out to about 100 plus days in milk before we started to see the percent of cases kind of mimic the percent of cows. So H5N1 does affect cows later in lactation. There are some reports of pregnancy being a risk factor, which would make sense. We haven't got to that point where we can say that looking at our data yet. So what impact does it have on total milk production and somatic cell count? This is just one farm. We've got about 25 of these, and they kind of run the gamut from almost no changes in bulk tank somatic cell count, less than a 5% decrease in daily milk production. This farm happened to drop about 15% in milk production, and their somatic cell count was up 126% from a 30-day baseline prior to the onset of clinical signs. We've seen farms that have been 300% of normal cell count. Usually those come back down after about a month. Some farms still struggle with cell counts out about 2 months, where they're kind of jumping around still higher than normal. So definitely impacts milk production and cell count, but it is variable farm to farm. So when we talk about disease spread risks, we know there are known risks in terms of lactating cattle. That's how this virus moved from Texas, Kansas to almost every other state that's become infected. People, I think, are a known risk, and this is kind of how I got involved in this when we had poultry facility in Michigan. It was one of the first ones that got infected with the dairy bovine strain. And what we found is there were people from the poultry facility that were working on the dairy on the weekends. And we presume that they're probably carried the virus back to the poultry facilities. Assumed risk, service personnel and vehicle movements. And especially as these dairies are getting larger, and the large dairies, they have people moving on and off the farm essentially all the time, and could those be bringing the virus with them? As far as assumed low risk, feces is assumed to be a low risk. We've done a lot of PCR testing of feces looking for the virus, and haven't really been able to detect it. Manure, which is a combination of feces and urine, we do know that it shows up in urine occasionally, but not real frequently in cows. So could lagoons be a source of disease spread? Could spreading manure on fields be a source of spread? And there were definitely some poultry farms that had dairy manure spread on adjacent fields. That's how the poultry facilities assumed they became infected. I don't think we have any really good evidence to support that at this point. As far as low risk, I'll put milk trucks on there, and then we'll talk about the caveat. Migratory waterfowl, low risk until probably late last year, early this year, when we had 2 additional spillover events in Nevada and Arizona with the D1.1. And then peridomestic birds, so pigeons, starlings, sparrows, birds that hang out at dairies a lot. So there's, and we'll go through the milk truck testing, but part of the problem is that we got into the milk trucks too late in the outbreak in Michigan and California both. And so we don't know really how much contamination of those trucks and potential spread was happening during the peak of the outbreak in those states. The peridomestic birds, a lot of the testing that's been done on that has happened, again, a couple weeks after onset of clinical signs in those herds, and usually what they find is less than 5% or so of those birds are infected, and most of them look like they're a spillover from that dairy cattle herd, not necessarily the source of infection for that herd. So I'm gonna put a question mark at the low risk. I'm not sure we know what kind of risk these really are. So other than the lactating cattle, which we know are a proven risk, I think there's a lot of other risks that are probably low risks, but are still ending up in spread of the disease. And that's what makes it, I think, tough to come up with what biosecurity practices should we implement. Well, if you can help it, don't move any cattle. If you can help it, don't move lactating cattle, especially bring them onto your farm. After that, all the normal biosecurity practices are good, but I don't know if they're going to have any impact in terms of stopping the spread of H5N1. So how can cattle be exposed? Through the ocular, through respiratory, through bites. There's been some interesting work done in California with flies, where they found flies carrying H5N1 at pretty large viral loads. And if these flies can transmit it is still unknown, but we have plenty of flies on dairies that could move from dairy to dairy and spread the disease. Rectal and vaginal, I know there was some vaginal swabs that were done in Michigan. I believe all those were negative, looking for the virus. But when we were doing urine sampling, which could contaminate the vagina, we were finding occasional urine sample positives. Oral through feed and water, feet, injections, and then teat and udder. And teats really where I think initially this started. Is this is a disease that's being spread in the milking parlor. It's essentially being spread like other contagious mastitis pathogens, Staph aureus, for instance. And when we talk to dairy producers, a couple things in the USDA questionnaire, which I was initially involved in working with, we asked about the milking order to try to see if it was following, does the spread of disease, clinical cases kind of follow the milking order? And a lot of producers said it wasn't following the milking order. But the other thing that I thought was impressive about it is it seemed to be moving much faster than what we would typically see Staph aureus or mycoplasma, strep ag move through a herd. So what we were told is it's in the milk, and you really need to focus on that area in terms of disease spread. And so it would make sense that if we have poor milking procedures, people that have a lot of liner squawks, air admission, and they're getting reverse jetting of milk up into quarters from the previous cow, that they could be spreading it if they're not doing a really good job of post-dip milk, teat dipping, those kind of things. But this disease really didn't spare very many farms in an area, so I don't really think that it's primarily spread through milk on dairy farms. When we looked at dairy environments where we sampled and looked for milk exposure, we did find that yes, most of the detection of influenza A virus was in the parlor, through parlor surfaces, through milking equipment, just because of the contamination of the milk. We did find it in some wastewater and lagoons. Most of those were collected in Michigan because of the concern about spreading manure close to poultry operations. Other than that, we really didn't find very many. This was, I believe, 600 samples that we collected on multiple farms. The picture on the right is kind of interesting. That was a farm we went to take environmental samples from, and we took a sample of the milk that had been spilled in the driveway, and sure enough, it was positive for virus. And there was some kind of equipment malfunction within the milk house, and there was milk all over that milk house, and then it'd come outside. So definitely in the milk and around the milking environments. So as far as vehicle testing, so milk tankers, we do have milk residue that comes out of this vent at the top. This dam is supposed to capture it and come down this down tube, but a lot of them aren't necessarily functional, and we have milk coming over the sides of the tanker. We tested, swabbed those. We tested tires on this bulkhead unit. We tested the dipper and swabbed inside there, swabbed the hose, and then on these farm load we did the same, where we tested pretty much everything that we could find. Some of these tankers did have raw milk present at the time of sampling. So in Michigan, we sampled in June of 2024. We sampled 18 milk trucks, over 112 samples. We also sampled within the cab of the truck, and we asked the drivers to walk through a foot bath where we could potentially capture the virus. All of those samples from the exterior and the interior of the cab of the truck were negative. We had one positive detection, and that was actually on this direct load cabinet base here, which was kind of interesting because we thought that, you know, especially the farm loads that have the hose that goes into the milk house, and then you go to the next load, next farm, pick up another partial load. That's where we were potentially going to have more contamination. On a direct load where the hose is associated with the farm, we really didn't think we were going to have a positive. Did find it on a dead stock trailer and a livestock trailer. That was on the infected premises, so those trailers hadn't moved off when we tested them. And then we went to California in April of this year. We tested 20 milk trucks. We took 119 samples. We weren't able to get milk from inside the tanker in Michigan, but we were in California. And the one positive detection out of those 119 samples was milk from inside the tanker. No external contamination that we could detect with virus either. And we also did the cab of the truck as well. So it looks like really low risk in terms of milk trucks, but the caveat is that Michigan had their outbreak in April. Primarily California was January. It started before that, but that's when it was in this area. So we were a few months behind really the peak of the outbreak when we finally got to testing vehicles. So one of the things that we've been concerned about is aerosol transmission. This is an influenza virus. A lot of them are transmitted through the respiratory route. We know cattle don't have as many of the influenza receptors in their respiratory system as other animals. But it kind of made sense. And in February, this, just because of the speed of the spread throughout the herds, and this is an article that came out in February from the Czech Republic evaluating the genetics of the virus and then the meteorological conditions and determining, okay, could this have happened? And so in this, they had 3 poultry farms, B, C1, and C2. They went through the genetics, and they were definitely essentially connected. And so they looked at the air intakes of where these farms would allow air to come in, where air would come out. They looked at the landscape, they looked at temperature, and essentially their conclusion was, yes, it is potential for aerosol or wind movement to have moved this virus from one farm to another. So we've been taking air samples in Colorado last summer, and we took probably over 100 air samples, and we weren't able to detect virus in any of those. A couple of the ways we tested here, we test individual cows. Essentially, this is just a little vacuum pump that goes through a filter that would capture the virus. When we were collecting milk samples in the parlor, we had a little filter here so that essentially what the workers were getting exposed to is what we were trying to sample with our samplers. So with our collaborators from Emory University, we were able to detect virus in the air in milking parlors and from exhaled breath of cows. Not many of those samples were positive, but we did find it. Kind of reminds me of the peridomestic birds and how only a few of those we find virus in, only a few of the milk trucks. We don't have anything that's really jumping out other than lactating cows. They also found infectious H5N1 virus in the air and in the waste or water stream. So usually when we look for it, we say, yes, it's present. With our PCR, polymerase chain reaction testing, that doesn't tell us if it's alive or dead. That just tells us tells us that the RNA or the DNA are present. In this experiment, they actually went ahead and injected that and found out that yes, it was still capable of infecting animals. So another thing that kind of drew us to this aerosol investigation was a collaboration we had with Lander Vet Clinic in California. They enrolled 19 farms before the disease was in their area, and they were doing daily bulk tank samples from all these farms. And farm BC and BO broke in late October. And then we went about another month, and then the other 17 farms all broke within about a 3-day period after a bomb cyclone went through. And so one of our thoughts is, you know, did the air move? Did that move flies? Did that move peridomestic birds? Did that move essentially the virus could be in aerosols with milk fat that would help protect it? Could that have moved the virus up into all these farms? So one of the things that we did, just for a start, we started with that farm BC, our first farm, which is the red dot here, and we asked our friends at the Atmospheric Sciences Department here at Colorado State University to do some wind modeling for us. And so if we go back 3 weeks before BC showed any clinical signs, and all these lines essentially are converging on red. So that means all these coming from the left are essentially out of the northwest. These yellow coming from the blue dot, which is our potential source farm, toward the red dot, we're coming out of the south and east. So 3 weeks prior to BC becoming positive in their bulk tank, we had a few winds coming out of the right direction. 2 weeks before, we didn't have any winds. All the winds were really out of the north and west. And then the week before, we had quite a few winds that were coming out of the right direction that could have moved it from the source farm to BC. So our goal now is to get whole genome sequences of these farms and start trying to pair them up and see if we can at least have any evidence that wind could have been a factor in moving, like I said, flies, birds, or just aerosols from one farm to another. So when we were in California, as I mentioned, we were doing bulk tank sampling. So prior to infection, and then as soon as we detected it in the bulk tank, then we went out and we did some testing on those farms. And before I go any further, I want to just point out this PCR and the CT values. You might have heard that. What essentially a polymerase chain reaction is it's amplifying the sample to try to find the virus. So we're trying to get the virus, we're essentially replicating the virus as it goes. And so the lower the CT value, the higher amounts of virus, which means it didn't take as many cycles to find it. And so this is what it looks like. A CT of 40 is essentially 0. A CT of 13.5 is 40 million copies of RNA of that virus. So it's a logarithmic or an exponential increase in the amount of virus based on the CT, but it's backwards to what we typically think in terms of the lower the CT, the higher amounts of virus. And the presence or detection doesn't mean the pathogen's viable or infectious, like I mentioned. It's just telling us whether there's DNA or RNA in this case in that sample. So this is this was BC that we followed. So here's they started back in early October, followed it. They were negative for a period of time. The first bulk tank detection of the virus was on 10/24. They noticed nasal discharge, such as this cow, on 10/28. We were out there on 10/29 collecting samples. And what they did is once a few of these farms were positive on the bulk tank, then they went to individual pen sampling. And I'll show you what that looks like. It's kind of messy here, but essentially 11/1 was the first date of clinical signs on this farm, and you can see it's already in half of the pens. By the time we see any clinical signs, so I know there were some recommendations that quickly move cows into the sick pen, which you should do if they need treated and have any withholdings anyway. But you're probably not going to have an impact on disease spread because by the time you start seeing clinical signs, it's already in half of the pens. If we follow this out over a period of time, one of the things that was interesting to us is this farm is segregating sick cows, moving them to the sick pen, and their individual pen samples all still mimic the bulk tank. And so this kind of threw us a little bit for a loop as why are we seeing this? We assume that cows have CT of 20 is a lot of virus, especially for a pen sample or a bulk tank sample. Why aren't these cows sick? Why are we having this much virus in that milk? So that was one of the questions. And when we started looking, and this is the same farm, BC, and I'll go through the study in a minute, where we enrolled 20 clinical and 10 non-clinical cows that were presented to us by producers. This was cow 58536. Blue is her milk production curve. And essentially, she was sitting at 100 pounds of milk. And when we look at the CT value, she was essentially negative, and then she dropped to 19.3, which is a lot of virus being shed in the milk. And she was positive, or at least we detected virus in her milk at 6 different samplings. This cow never had any clinical signs, never showed any milk drop. And when we started looking at more of the non-clinical, because initially we're like trying to figure out the clinical cows and what's going on with them, we found out that there's a lot of cows that are walking around perfectly, look perfectly healthy, giving an amazing amount of milk and shedding a lot of virus. And so that's why we have CT values for 20 of all pens. For this particular herd, we've got this cow that's essentially giving 100 pounds of milk at a CT of 20. And like I said, this is an exponential increase in CT in the amount of RNA. So when we get down into 15, we're talking about that 40 million copies. So at a tanker level, this one cow would, with her 100 pounds of milk going into 50,000 pounds of milk, would still be positive on a PCR test in the upper 20s, approximately, based on the calculation. This herd was 3,000 cows. They were producing 250,000 pounds of milk a day. If we had a single bulk tank sample for this herd, we had multiples. If we had a single bulk tank, it would probably be in the low 30s, so still detectable. So what we found is that one cow that's shedding a lot of virus, giving a lot of milk, looks absolutely normal. You wouldn't be able to go ahead and pick her out as a sick cow. She can be responsible for your entire bulk tank being positive for 3,000 cows. And so now we're saying, okay, now we're starting to be able to figure some of these things out, why we're seeing what we're seeing. And that non-clinical cow shedding a lot of virus was kind of critical. So the other study that we did on some of the farms in California, but we did it on 8 farms in Colorado, was looking at shedding routes and duration via longitudinal sampling. So where are cattle shedding the virus? Nasal respiratory secretions in the urine. We stopped collecting feces because we really couldn't find it there. We didn't sample saliva necessarily, but we did sample a lot of milk, and milk was the primary place where we found it. This is just a map that shows where we sampled. So 3 farms in California, 8 in Colorado, and we did sample the farm A in Ohio as well, with cooperation of USDA. This is our sampling specifics. So we looked at a lot of different cattle types: pre-weaned heifers, dry, fresh cows, clinical, non-clinical, and then we took those environmental samples. But I just want to really talk about the clinical. So the plan was to enroll 20 clinical cows, 10 non-clinical. Those we just asked the producer to provide those for us. We didn't go looking at records trying to determine who was clinical and who wasn't. So we did nasal swabs, blood, serum, and urine once a week on those cows, and we did milk twice a week, and we followed them for 10 weeks. During this time, when we were collecting serum, we actually found viremia in 20 of the cows in 3 herds in California. And this was important because in experimentally infected cows, where they'd looked for viremia, they hadn't found it. But we had a lot of questions when we're finding virus in urine. How does virus get to the bladder? How does it get in the urine? And really, through a viremia, where the virus gets into the bloodstream and moves throughout the body, is really the only way that it makes sense that it would get to the kidney. And so now we were able to explain that the reason it's getting in the urine is because these cows have this viremic event, and also explain some of the aborted fetuses. We did have one of the farms that did have a viremic cow. They did have an aborted fetus that was high path positive. And since then, we've heard a lot of abortions in California that have been PCR positive for this virus. Interestingly, when we were in Michigan, we were working with a farm. We submitted, I believe, somewhere close to 10 fetuses. They had an outbreak during their clinical signs in their cows, and none of those were PCR positive for for the virus. So not sure why the difference, but they didn't have near as much trouble finding it in California as they did in Michigan. But that also brings up some concerns about food safety. If we've got virus in the blood, that means essentially it could go to most organs in the body. FSIS, USDA looked at condemned carcasses, and they've only found it in one. I believe they found it in the diaphragm and in the kidney in one cow. And then fetal serum products, if those are being used in laboratory testing, need to make sure that those don't have H5N1 in them. So as far as our longitudinal study summary, I could show you individual cow information. You'd probably get pretty bored of that pretty quick. I can tell you that some clinical cows did not have virus detected during our sampling. So they didn't have it in nasal swabs, they didn't have it in milk, they didn't have it in urine. Alternatively, we did have some non-clinical cows, as I've already showed you, that we did have virus detected. Milk is the most commonly PCR positive sample we collected. About 60% of the clinical cows that were presented to us and about 18% of the non-clinical cows that we tested had virus in the milk. Urine and nasal swabs, not commonly positive, less than 10% of the cattle we tested. And it was similar in clinical and non-clinical cows. The clinical cows did not have a higher percentage of urine or nasal swab positive. So whether they showed clinical signs or not really didn't predict whether they were going to have it in their urine or nasal swabs. And then serum, also not commonly positive. California herds are the only herds that we've worked with that really looked into the viremia. 16% of the clinical cows and 3% of the non-clinical that were sampled were positive in the serum. As far as how frequently and how long do we find viral RNA in these samples, a lot of cows will have it just in one sampling, and we were sampling twice a week for milk and once a week for the other samples. So some cows we'd only find it one time. We did have some cows that we found it every sampling up to 89 days. And this kind of brings up the concern about, do we have some kind of chronic carriers of high path, and are they going to continue to expose naive animals, first lactation animals, as they're introduced to the herd? So viral RNA detection doesn't always coincide with clinical signs in these herds. So this is just a couple herds showing you some of the antibody results. So not looking for actually the virus, but looking for the cow's or a cattle's response to that in the terms of antibody production. 92% of all the cows that we enrolled on farm AO had antibodies, 100% of the cows on AX. That tells us that all the cattle are exposed, even though only maybe 10 to 20 to 30, maybe if you're in California in the heat, 50% of the cows that show clinical signs, all the cattle are exposed. A lower percentage of fresh cows than lactating cow. This should be all lactating enrolled cows. Fresh cows, 43% had antibodies on AO, 73% on AX, and then 29% of the dry cows had antibodies on AO. We did see some difference in pre-weaned heifers, and maybe some of that was due to management. AO they have a separate calving and heifer raising facility compared to AX. Only 7% of the pre-weaned heifers on AO had antibodies, 87% on AX. And those calves were bled about 4 to 6 weeks of age. So is offsite rearing reduce exposure? It looks like it might. Is that a good thing, or did these pre-weaned heifers essentially get their first vaccine? That's really, we'll have to wait and see what that looks like. So one of the things that we've been doing, and we're actually doing today, is collecting DHI milk samples from 6 herds in Colorado. So we get all samples from all their lactating cows, and we test those for antibodies. And so this is one of the farms that we worked with, and shows the percent of cows within that lactating herd that have antibodies. This is kind of an unusual one. It has higher antibody levels than normal, but we look across all lactations, it's running between about 40 and 45%. First lactation, not really a lot of difference in antibody level as we go from first, second, third, fourth lactation. They all look pretty similar. I think what's interesting about this farm, and this is the green bars represent 407 days since the onset of clinical signs in that herd, is this herd we started working with them a couple months after the outbreak because they had some chronic cows, and not only chronic, but they had new infections coming, and they're trying to figure out, what's going on. But we're trying to figure out, and we just got these data back last week, is why they jumped up about 10% in the percent of antibodies in some of these classes of cattle at their last milk test. And are these cows that were clinical at the time that have maybe they were dry during that 315-day test and now they're coming back, or what is responsible for this increased level? Most of the herds that we're looking at don't have that peak. They have declining level of antibodies in their lactating cows, which means we need to have a vaccine coming. So, summary of the longitudinal summary in terms of antibodies commonly detected in the serum, 90% of clinical and non-clinical cows. So almost all cows are exposed whether they show clinical signs or not in the lactating herd. 60% of fresh cows, 30% of non-lactating, and then 20% of pre-weaned heifers. So duration of antibodies in cattle is still ongoing, but we do see it in herds after 400 days. At the herd level, though, less than 50% of lactating cows have antibodies 1 year post-infection. What we're told is for herd immunity to work, meaning the herd to be protected from a new incursion of disease, we need 90+% of the cattle to have antibodies, and we're not seeing that any of the herds we're working with, and that makes a lot of sense. Our doctors ask us to get vaccinated for flu every year because the duration of immunity isn't that as long for the flu virus. So we really need vaccines if we're going to protect our herds from future incursions of disease. Lastly, I just want to thank my research team here at Colorado State. I'd only been here 6 months. I just moved over from USDA when this happened, and so it was really good to start getting folks on board that could help. A lot of these people were recruited from USDA after they retired. So acknowledgments. The first one up there actually should be dairy producers. Really, none of this would have happened without the cooperation of dairy producers in the states we worked in. And this was a disease issue that veterinarians weren't going to solve alone. Epidemiologists weren't going to solve it. Regulatory officials weren't going to solve it. Dairy producers weren't going to solve it. We all needed to work together to try to figure out what's going on. So thank you for all the dairy producers out there that opened your doors and had us gave us access to your cows so that we could collect these samples and come up with the data that we have. Thanks to the CSU Veterinary Diagnostic Lab and the Iowa State Veterinary Diagnostic Lab, they've tested somewhere in the neighborhood of 50,000 samples for us over the past 16 months. The Colorado Department of Ag, the California Department of Food and Ag, CDFA has been an amazing partner in all this. To work with as well. The Dairy Authority, where we're at today, collecting milk samples, the Atmospheric Sciences Department and the CSU Centroid that are helping us with cattle movement tracking. So I think with that, I'll try to answer any questions. I'm going to go ahead and escape out of this so that I can see the bar with the chat.
Thank you, Jason. That was a great presentation. If you want to go to that last slide, if you would, please. The very last slide on your, there you go. Thank you. To view this webinar or any of our other webinars, you can visit our archives at any time at www.hordes.com/webinars. We hope you'll make time to attend our next webinar, which will take place on Monday, October 13th. At noon Central Time. Pam Rug of Michigan State University will be discussing using key performance indicators to manage milk quality. Then in November, we'll discuss a feed and forage outlook. So now we had some questions come in. First one is, if the most recent infections in dairy cattle were the result of contagious activity among livestock, is the fall wildfowl migration a concern?
Yes. I think that's at this point right now where it seems to, like I say, no new infected premises in the last 30 days. I think that's really good. I think the risk, there's two risks right now. One would be the wild migratory birds coming back. And even though we've only had 3 known spillover events, it's interesting when you go start talking to veterinarians in all these states, and I've had veterinarians in Michigan, Colorado, New Mexico, California, Minnesota say, we've seen this before. And and since 2022. And it seemed to kind of die out. And a lot of those producers said we had dead cats. And granted, it could be something else, but it kind of makes sense as much influenza A was floating around in the wild migratory birds, how many commercial and backyard flocks were becoming infected. It would be completely feasible to me that this would get into a herd or two and kind of burn itself out. I think it got into the right population of a dense cattle down in Texas and Kansas area, and then through cattle movements it kind of spread. We saw what it did in California in terms of it just burned through there like wildfire. So I think that wild migratory birds are still a concern. And then the other concern are some of these herds may have some of these chronic carriers that are still shedding the virus at low levels. And I was on a 10,000 cow dairy a few months ago that was still bulk tank positive. And I was like, how many cattle do you want to test to try to find the 2, 3, 4, 5 in your herd that could be shedding the virus? And so I think that cattle movements are still a risk. But right now, it seems like the wild migratory birds are probably on the radar.
All right, well, you kind of answered this next question a little bit in your last answer, but what herds tend to have an H5N1 recurrency?
That's a really good question. We've been so busy testing and trying to do that we haven't dug into some of these herds. And I could show you some bulk tank results where the bulk tank goes negative on most herds about 30 to 45 days after clinical signs. Kind of most herds don't know when their bulk tank first went positive. They know when they first saw clinical signs. But we do have herds that are out 70, 80, 90 days and still have positive bulk tanks. I know of one herd in particular that was positive for over a year, and trying to figure out what's going on in that herd. I think that it's part of that's got to be naive animals coming in to the herd. And I think the other thing would just be chronically infected cattle that just keep shedding it. And that's going to be a tough thing to find in large herds.
All right, next question is, what might be the reason that Washington has been H5N1 negative so far?
Well, I don't know specifically about cattle movements into Washington. I'm going to say that I think the federal order in testing cattle helped a lot with preventing state-to-state spread. To be completely honest with you, I think luck has something to do with it in some cases, both from their exposure to wild migratory birds and from cattle movements. But I think that states that didn't have it, that have large dairy populations, have hopefully dodged the bullet. Why they've dodged the bullet is really unknown. And I think we may never figure out exactly how this is moved from farm to farm. So I said cattle movements is how it moves from state to state. Once it gets into an area, it seems to move fairly easily. And last I saw from USDA, 50% of the farms, almost 50% of the farms, didn't have any cattle movements that got infected. So it's moving other ways, and trying to figure out what those are has been a challenge. And I think producers get frustrated when they're told to do 50 biosecurity practices and then they don't work. And that's kind of what pushes us toward aerosol being a potential route of transmission, because you can't stop air movement.
Yeah, that makes sense. Even Wisconsin has been in that situation as well. Next question that came in: although antibodies might not be detectable after a year, could they still be protected when challenged?
That's what we use kind of as the standard to looking at it. Could they be protected? I don't think that at the levels we're seeing it, with 20-30% of the lactating herd having antibodies, I do not think they would be protected.
That makes
as
sense.
fast as we have some dairy turnover, I mean, a third of the herd wasn't lactating last year when this disease came through, and if they were housed off-site and away from potential exposure, you've got 2 years of animals potentially that are going to come in without any prior exposure, and even if they were exposed 2 years ago, they wouldn't probably have any persistent antibodies.
Makes sense. Another question that has come in about why, let's see here, any explanation why dairy herds in the Northeast are not positive for H5N1, so similar to the other states, and also why beef herds are not positive?
I was wondering if that was going to come up. So I think part of it is cattle movements. There just aren't as many cattle movements to the Northeast as there are in the West. So I think the risk of moving the disease was a lot lower in those areas. I still think luck in some cases comes into play. Beef herds are really interesting because as we're trying to figure out how this is moving from herd to herd, and we have beef herds right next to dairy herds, we have tested somewhere in the neighborhood of 150 beef cattle in Colorado and California, usually pretty close to dairy herds that were infected. And we cannot find antibodies to this virus in those beef cattle. And it makes no sense to us. We've had some feedlot cattle we tested that the manager who's been feeding cattle for 20 years, he sent me some pictures of nasal discharge that was abnormal to him, which is what we're seeing in the dairies. The cattle were actually some of the best gainers he's had, but we did the testing, and we do find positives, but they fall into that percentage that could easily be false positives. So why we're not seeing it in beef cattle makes no sense to us, and we'll keep testing, but we're kind of out of areas. There's not any hot areas right now, so we don't really have anywhere to look. And most of the animals, other than that feedlot herd, did have sick animals around the time the dairies were sick. Dairies had clinical cows. So we thought that this would be an easy thing to prove, is that, they are bovines. There's no reason they wouldn't be susceptible, but through our testing, we haven't been able to detect that.
It's very interesting. Question along the lines of how it jumped, the spillover, as you said. How can a virus that infects poultry or wildfowl is adapted to their tissues? How can it make the jump over to cattle?
How it jumped is not really known. I mean, we have birds on dairies all the time, and some of those could be carriers from the wild migratory birds too, because they interact with them. When this first started, and we heard about a large number of bird die-offs in the Southwest, in Texas, around some of these dairies, my understanding is some of those dairies were green chopping at the time, and somebody said, well, those cutters might be 18 inches off the ground. They're not going to suck up a dead bird, or they're not going to hit a dead bird that's on the ground. But my guess is that that's one of the potential routes that I heard of. And it would make sense to me that there's enough vacuum being kind of produced from green chopping that you could suck some dead birds in and then feed it directly to the cattle if oral exposure would lead to disease. Or whether it was contamination of other feeds or water. I have my brain can't put together how a virus gets from the bottom of a shoe or something like that into the teat of a cow. I guess it could have come in through some, contaminated bedding at some point. But I think that maybe some oral or respiratory would have been the first route of exposure for some of the cattle, and then it kind of got into the milk. And once it gets into the milk, so a milk has a lot of the receptors for flu in it, and the mammary gland is such an active metabolically active gland that it can just crank out so much virus because it's cranking out so much milk. It's just a factory to do that. So it seems like it found the perfect host to kind of propagate in milk. And then the question is how is it moving from cow to cow via milk? And so I think there are a percentage of cows that are infected through the mammary gland, through the milking process. Interestingly, when studies have tried to replicate that, they haven't been able to move it from cow to cow with a contaminated milking machine. They've dipped teats in contaminated milk in naive cows, and they can't get the disease to spread. The one study that actually opened up a lot of answers for us too is they didn't have anything they could transmit it to these naive cows, so they dumped milk over the feed. Those cows developed antibodies, never got sick, and then were essentially immune when they tried to experimentally infect them with intramammary challenge. And so that's what I think we're seeing in some of our cattle, where we see a PCR positive detection in milk, but the cattle are never necessarily sick, because they were exposed through another route and developed antibodies and then are protected.
That's really interesting. Wow. We have 2 more questions that will get answered, and then we'll wrap up if that works for you.
Yep.
All right. Has there been any genetic analysis done for cows that are more resistant to the virus?
That I know ARS, Agricultural Research Service, working on, and that reminds me that I have some samples to get to them. We didn't have a lot of herds that were doing genomic analysis, which is kind of interesting that we worked with, but we do have one herd. So no, we don't have any of those results, but yes, we are looking into that through our friends at Ag Research Service, USDA. And there may be other folks that I'm unaware of that are doing that as well.
Excellent. And then the question that is very popular, what is the status, or do you know the status of the vaccine availability?
I think vaccines are really close and maybe even ready to be implemented. My understanding is it's as much, the efficacy is probably there. It's in the production. I think it's the regulatory. So I think it's approval, and I would hope that we have them sooner rather than later. What that means, is that 3 months? Is that 6 months? Is that a year? I hope it's sooner rather than later. But when we get in this part of the process of vaccine approval, everything is really quiet until everything is completed. And so you don't get much information at this point unless somebody's going to put out a press release that something's going to be released soon. But it's well on its way, and I just don't know the timeline when it would be primarily approved. I think the product is there. I think we'll have good efficacy of the vaccines that I've seen some of the preliminary data for, but availability, I don't have an answer.
Well, that makes sense. Well, that wraps up our questions. So thank you very much, Jason. And to our listeners, thank you. We appreciate the time that you spent with us today, and we hope to see you in the audience at a future webinar, or tune into our Hordes Dairyman podcast on YouTube or your favorite podcast player. Until next time, goodbye from us here at the team at Hordes Dairyman.
Thank you.
Thank you.